Okay, continuing our discussion of fertilization in implantation and embryogenesis, focusing on the second one of those now. This is lecture 14B on implantation. So the blastocyst has now reached the inner part of the uterus, right? It's now in the uterine cavity and it has differentiated. We have that inner cell mass and we have the outer trophoblast. And what's going to happen is the trophoblast, this outer cell layer, let me pick a different color here, right? The trophoblast here, that outer ring of cells, is going to produce some enzymes that allow it to burrow down into the endometrium. So this entire area that we're seeing here is all endometrial lining. So the blastocyst isn't just going to attach to the endometrium, it's actually going to completely burrow down within it and kind of pull the covers over its head, so to speak. So the trophoblast makes enzymes that allow it to kind of eat away and burrow into the endometrium and then start sending out fingers of tissue, fingers of groups of cells called villi. They're going to start attaching into that endometrial lining. Here's the text if you need to pause and catch up on that. Once it does this, once the trophoblast has implanted into the endometrial lining, it's going to start to secrete HCG, which stands for human chorionic gonadotropin. HCG does several things. It's going to tell the corpus luteum, remember, that formed from the empty follicle after the oocyte was ovulated and was producing progesterone to make the endometrial lining nice and thick and juicy. HCG is going to tell the corpus luteum, hey, we got one. We're going to try to get pregnant. Don't stop making progesterone. So that way the corpus luteum continues to make progesterone and not degenerate, because if it degenerates, progesterone levels fall, and then you're going to lose the endometrium and the blastocyst along with it in a period. So HCG tells the corpus luteum to keep going, keep making progesterone so that the pregnancy can get established. HCG also sends some signals out so that the woman's immune system is less likely to attack the blastocyst, because the blastocyst is not her, right? It's a foreign invader in some ways, so you need to calm the immune system down so it won't kill it. HCG is also going to help stimulate the production of other hormones by what is the trophoblast, which is going to become the placenta. Eventually, the trophoblast placenta is going to make its own progesterone. So we're going to get that system starting, get that up and running. And HCG is the basis of pregnancy tests. So when you check to see if you're pregnant with one of those P6, or if you get blood drawn at a doctor's office, what they're looking for is the presence of HCG, because the only thing that will cause that, almost the only thing that will cause that, is a pregnancy. Now it's interesting to note that there is no production of HCG until implantation has occurred, which is usually somewhere in the first week to 2 weeks after fertilization. So we know then that if someone had sex, unprotected intercourse, 2 days ago, it's too early to do a pregnancy test. There would not be any HCG yet. So when we get to the end of the second week after fertilization, we've gone through cleavage, we've formed a morula, we've formed a blastocyst, which is now implanted, HCG is detectable, and the woman is considered pregnant. This is 2 weeks from the date of fertilization. However, the convention is that she is called 4 weeks pregnant. And this is because way back when people first started tracking pregnancy on calendars and figuring out when due dates were, nobody knew what ovulation was, and no one certainly knew when it occurred. But what they did know is the first day of the woman's last menstrual period. So they went by the first day of the last period, so it gave you that extra 2 weeks in most people, because ovulation usually happens at day 14 of the menstrual cycle. So we have a little bit of a discrepancy in terms of how we talk about pregnancy and what's technically been happening. So if you kind of look at this comparison, on the upper level here, we have menstrual calendar, which is the traditional way to talk about pregnancy, right? And so you would have menses here, and then ovulation, and then your positive pregnancy test would be, probably week 5 by the time you realized you were missing a period. If we count from day 1, week 1, by the menstrual calendar, a full-term pregnancy lasts 40 weeks. If we start from when fertilization occurred, however, everything is 2 weeks shorter, right? So we only have 38 weeks of a pregnancy. So you'll hear me talk sometimes about dates by menstrual calendar or dates by fertilization. So that's confusing. I apologize for that. So a lot of times people ask me, well, wait a minute, how do twins happen? And so this is the perfect time to talk about how twins happen. So first, let's consider the case of identical twins. Oh, they're so cute. So identical twins are identical because they have the same genes, they have the same DNA, they have the same exact chromosomes. So you know then that if you have the same DNA, you have the same chromosomes, you must have all formed from the same original cell. You must come from the same exact zygote. So the technical term for identical twins is actually mono, which means one, monozygotic twins. So then we have to say to ourselves, well, then how did you end up with 2 separate embryos? Well, for identical twins, for some reason, at some point, that little clump of cells or ball of cells must have split into 2 separate clumps or balls of cells, each of which then went to go on and to kind of form its own embryo and fetus and baby. And depending on the timing of when that split happened, you can see different types of variations. So what this is showing us is up here at the top. If you split when you're still at the morula stage, then you're going to have 2 pretty separate pregnancies. They're each going to then form their own blastocyst. So you're going to have your own placenta and amniotic sac, right, and be completely separate. If the split doesn't happen until you've reached blastocyst stage and just the inner cell mass splits in 2, you can see how these 2 babies are sharing the same placenta because they were in the same trophoblast. If it happens even later, then look, here they're sharing the same amniotic sac. And if it's very late in stages that we haven't talked about yet, then you can end up with conjoined twins. The old term for that is Siamese twins, right? Conjoined twins. What about non-identical twins? This is a famous pair of twins because we tend to be fascinated by skin color in our country, but these are twins born to these parents, and as you can see, they have very different genes and chromosomes from each other. They're both little girls, so they're both XX with their sex chromosomes, but they have very different genes for coloring and pigmentation. And so if you have different genes or chromosomes, that means you formed from 2 separate zygotes. So therefore, so they're called dizygotic twins because di means 2. So therefore, they had to have come from 2 different oocytes. For some reason at ovulation, 2 oocytes, 2 eggs, instead of just 1, were released. And each one of them ended up getting fertilized by its own sperm. So that's how you ended up with 2 completely unique combinations of chromosomes. All right, so we've already done fertilization. This mini lecture was implantation. So we have that blastocyst with the outer trophoblast, inner cell mass, burrows with its enzymes down into the endometrium and then starts secreting HCG to tell the corpus luteum, hey, we got one, keep making progesterone. That HCG hormone is also what we detect when we do pregnancy tests. So we'll pick up next time with 14C, which is embryogenesis.